Hello. We know that DNA polymerase synthesizes DNA. We have discussed about replication fork. We have discussed about the complex enzymes or the numerous enzymes that are present at the replication fork. So the whole complex should work as one unit. And then there is always this DNA, the lagging strand synthesis has to open up and then new ones should be stretched out. How is this complex phenomenon accomplished? And the replisome, that is the cumulative of all these proteins, how do they work in and coordinate the whole replication of both these strands, one continuously and one discontinuously? That's a complex process. I am Dr. PCMR, Assistant Professor at Shastra. We will try to have a look at the complex mechanisms. It's actually difficult to explain in words, and the animations would certainly be of great help to visualize how things happen. If not exactly, at least it will give us a fair idea about the things. So the one way DNA accomplishes about the direction of the replication fork and direction of polymerization itself is by bending the DNA as shown here. And that is something like a trombone. You call it as the trombone model, and trombone is a musical instrument that is played with movable this tube like thing with a bend. So how does the replisome organize, and how is this whole thing coordinated? The one that is shown here is of eukaryotic, but the best understood one is from E. coli or the prokaryotic things. The prokaryotic one is replisome, that is the cumulative of all the proteins that are organized and which work as a unit at the replication fork is referred to as the replisome. And prokaryotic ones are much simpler and eukaryotic ones are much complex. So they do consist of a large number of proteins and with numerous subunits as well. The ones that are important for the polymerization itself are the core polymerases. That is, we have a DNA polymerase, that is the one that synthesizes the DNA, and they do have several other subunits such as for proofreading, that is the 3' to 5' exonuclease activity. And they also have the clamp loader, and that is the sliding clamp loader. And here we see that this is a sliding clamp, and the one that is shown here is the sliding clamp loader. And then we have these DNA polymerases, that is they are polymerase 3. DNA polymerase 3 is the one that is associated with replication, and because of their speed and good enough accuracy, and the processivity is increased by the sliding clamp. There are all these three, the sliding clamp loader and the DNA polymerases are bound together by these tau proteins, which are much more flexible, and then they accomplish a lot of things. We will try to have a look at how this might probably work in this cell. And there is a fantastic animation that is there on YouTube, and we can all watch it multiple times to have a best understanding. Here is the DNA that will show up. This is the replisome that is organized here, and the replisome has this is the sliding clamp loader, and the blue one that is shown is the helicase. It is separating the two strands. And then we have the sliding clamp loader here, and this is the sliding clamp that has come. It is now going to pick up a new stretch of lagging strand synthesis for synthesis of lagging strand. And here we see something coming and joining to the helicase near, that is the RNA primase or the primer synthesizing one. You can see the sliding clamp is opened up and put around the DNA and then brought back, so that this is the DNA polymerase that is synthesizing the lagging strand. You can see now a new sliding clamp has come, picked up a stretch of the lagging strand DNA, and then that is taken over here. and this continuous process keeps happening. This happens much more rapidly than what we are imagining, and it's fantastic to see that it does it so fast and so accurately. And that is the best thing about the invention of or what evolution has come up to make it stable. And I mean, the loss of sequence is managed or minimized, and then still the propagation is happening. So there are that is how DNA polymerase probably works in the cell. And if we compare the prokaryotic and eukaryotic replisomes, there it's much more complex. Just the polymerase itself is complex. In prokaryotes, we have 3 of these DNA polymerases that are involved in replication, and majority of the replication during replication, polymerase 3 is the one that actually performs majority of it. And there is DNA polymerase 1, which is involved in DNA repair and also RNA primer removal. RNA primer removal can also be removed by RNase H. But DNA polymerase 1 is special in the sense that it has 5' to 3' polymerization activity, 3' to 5' proofreading exonuclease activity, but it also has 5' to 3' exonuclease activity, using which it can remove the primers and fill up the gaps. And then ligase will take over and seal the gap, the phosphate backbone. So there are also 2 other, that is Pol 4 and Pol 5, which are involved in repair, like DNA polymerase 2 and 1 as well. And they are involved in translation DNA synthesis, which we will discuss when we study about repair. Comparing these with the complexity of DNA polymerases that are available in eukaryotic system, if you see, there are numerous of those. It's not just 5. So polymerase alpha, then we have beta, which is involved in base excision repair, then gamma, delta, epsilon, theta, etc. Majority of the replication is done by delta and epsilon. And they also have, for lagging strand, it is the delta, Pol delta, and for the leading strand it is Pol epsilon. DNA polymerase alpha is something important because it comes along with the primase and then synthesizes. After the synthesis of primase, it synthesizes a little bit of DNA and then falls off. Its processivity is less, and that is taken then it is taken over by either epsilon or delta. Delta for the lagging strand and epsilon for the leading strand. That switching, polymerase switching, you call that a phenomenon. So basically what happens is that the primase and DNA polymerase alpha are a kind of complex. They come over and then synthesize a small stretch of RNA, and then it is taken over by alpha and synthesizes a little bit, and because its processivity is less, it falls off, and then it is taken over by here, because this is going to be the leading strand, it should be the epsilon. So it will be taken over by epsilon and it will synthesize the leading strand. On the lagging strand, same alpha and polymerase alpha and primase come together, the RNA is synthesized, and then we have a synthesis of little small stretch of DNA, and then there is polymerase switching, which is taken over by delta, and then the rest of the DNA is synthesized in this fashion. And we will discuss some of these enzymes, the remaining that are that are involved in several of repair mechanisms when we study about repair. But otherwise, essentially, we have more number of factors because eukaryotic systems have very large DNA, and they have to be much more regulated, and their cellular complexity is also very high. Therefore, we have many more proteins as depicted here. Here is epsilon that is synthesizing a small, the leading strand, and delta, that is synthesizing the lagging strand. A helicase is called as MCM, and there are several other proteins associated with the DNA, like that is the histones, which is a part of our next lesson. I hope you understood the importance of the mechanism, how the DNA polymerase actually or the replisome accomplishes the synthesis of DNA. See you later. Have fun. Thank you.